Hello friends, welcome back to my YouTube channel Journey with Vijay Kumar Shrivastav. Today I am going to present the topic maintenance of genetic purity in seed production. First we will understand what is genetic purity. Genetic purity is the true to type nature of the seed, like seedling or plant or tree from the seed should resemble its mother in all aspects. It should be identical to its parents. Genetic purity is simply defined as true to type plant seeds conforming to the characteristics of the variety as described by the breeders. This quality character is important for achieving the desired goal of raising the crop, either for yield or for resistance or for desired quality factors. To maintain the genetic purity during seed production, in 1953, Horn has suggested some methods to follow for the same. Like first one is use of approved seed in seed multiplication to avoid any drawback or quality problems. Second, inspection of seed fields prior to planting. It will avoid any issues of volunteer plants in mixing of seeds production. Third is field inspection and approval of the crop at critical stages for verification of genetic purity, detection of mixtures, weeds and seed-borne diseases. Because of this field inspection at critical stages, we can rogue out the different plants of types or undesired plant to avoid any admixture which can reduce the genetic purity. And fourth is sampling and seeing of cleaned lots for testing purpose and its purity verifications. Fifth, growing of samples with authentic stocks or grow out test. Means these samples are going for grow out test, which is the method of growing the sample seed and observing the morphological and reproductive characteristics to verify its trueness. In 1968, Hartman and Kester suggested some methods to be adopted and followed to maintain the genetic purity. First one is providing isolation to prevent cross-fertilization or mechanical mixtures. This isolation refers to the distance which is required to maintain between the seed production field with other species of same crop to avoid any contaminations during fertilization, crossing period, pollination and avoid any mechanical mixtures after harvesting. Second is roguing of seed fields prior to planting. Means before planting we have to check the field selection. There should be no any issues of volunteer plants, means pre-existing plants which were grown in previous season of same crop. Roguing refers to the removal of such plants. Third is periodic testing of varieties for genetic purity. Whatever seed stock is there, we have to verify the genetic purity to avoid any contaminations or any impurities. Fourth is grow in adapted areas only to avoid genetic shifts in the varieties. The variety is released for the suitable areas for its characteristics maintenance, so it should be preferred to grow in suitable conditions, because in adverse conditions there are chances to breakdown of like mutations or some other problem to be arised due to genetic shifts. Fifth one is certification of seed crops to maintain genetic purity and quality. There is need of certification for its purity and it should be checked and verified on periodical basis. Sixth is adopting generation system. Means whenever we are using the seed in its production, there should be follow up to it should be maintained up to the permissible generations. There are various measures which can be used and followed to maintain the genetic purity of seed. Like first one is control of seed source or use of approved seeds in seed multiplications. Second, inspection and approval of fields prior to planting to avoid any voluntary issues or mixing during sowing. Next is providing adequate isolation to prevent contaminations. During seed fields and other fields, there should be a minimum distance to be followed to avoid any contamination chances. Next is seed multiplication outside their normal cultivation area to avoid any admixture issues from other varieties of same crop. Rogging of seed fields at critical stages. It's depend upon the crop, where the rogging removal of off types should be done on critical stages like vegetative stage, pre-flowering stage or flowering stage, and during maturity and harvesting stages. Next is periodic testing of varieties for confirming genetic purity. Certification of seed crops to maintain genetic purity and quality of seed. There should be frequent field monitorings till all operations getting completed to avoid any admixture or proper maintenance data recording of the seed production fields. After that, grow out test for verification of the seed produced. Now to study safeguards for maintenance of genetic purity. The important safeguards for maintaining genetic purity during seed production are defined as: first one, control of seed source; second, preceding crop requirement; third, isolation; fourth, rogging of seed fields; fifth one, seed certification; and sixth, grow out test. We will study in details about each methods. First one is control of seed source. The seed used should be of appropriate class from the approved source for raising a seed crop. There are 4 classes of seed from breeder seed, which are given and defined by Association of Official Seed Certification Agency. First class is nucleus seed, which is a handful of seed maintained by concerned breeder for further multiplications. The nucleus seed will have all the characters that the breeder has placed in it, and it is of highest genetic purity. The quantity of nucleus seed is very small in kilograms. Second is breeder seed. This is produced by the concerned breeder or a sponsoring institute, and which is used for producing foundation seed. It is of 100% genetic purity, and the label or tag used for breeder seed is of golden yellow in color. Third class is foundation seed. It is produced from breeder seed and maintained with a specific genetic identity and purity. It is produced on government farms or by private producers, and the quality of foundation seed is certified by certification agency or private companies. The certification tag or label issued for foundation seed is of white in color. The fourth class is of registered seed, which is not in practice in India. Here we will go through classes of seeds and its multiplications. There are 4 classes. First one is nucleus or breeder seed, which is developed by the breeder, and it is taking production on experimental stations directly under supervision of breeder. In case of nucleus and breeder seed, the highest genetic purity is maintained, and this is the source of all classes of seed. For this, the tag color is used golden yellow color. Second class of seed is foundation seed, and the breeder seed is source for foundation seed production, and this is produced by the National Seed Corporation or state seed corporations on their farms or by the private seed agencies. And this foundation seed is known as mother seed also. And in this case, the genetic purity is maintained as per the crop specific parameters, and breeder seed is the source of foundation seed. In case of foundation seed, the tag color is used white. Third class of seed is registered seed, and foundation seed is the source of registered seed. This registered seed is omitted in India, and it is not in practice. In case of registered seed also, the genetic purity is maintained, and the tag color used is purple color. Fourth class of seed is certified seed, and foundation seed is the source of certified seed, and the seed genetic purity is maintained as per the standards, and the tag color is used blue color. Second safeguard measure is preceding crop requirement. This has been fixed to avoid contamination through volunteer plants and also the soil-borne diseases. Volunteer plants are the plants of the same kind, same crop growing naturally from seed that remains in the field from a previous crop. Volunteer plants are plants that grow in the fields from the seeds found in the soil for same crops from previous season. These seeds should have been fallen from previous crop as they germinate without sowing or they volunteer to grow by themselves, so the name is given volunteer plants, and these volunteer plants should be rolled out timely from the seed field. Third safeguard measure is isolation. Isolation is required to avoid natural crossing with other undesirable types in the fields and mechanical mixtures at the time of sowing, threshing, processing and contamination due to seed-borne diseases from nearby fields. Protection from this source of contamination is necessary for maintaining genetic purity and good quality of seed. Now to understand isolation distance. Isolation distance is the minimum separation or distance required between two or more varieties of the same species of the crop for the purpose of keeping seed pure and avoid contamination. In hybrid seed production, if there is female contamination with foreign pollen or physical admixture, it will result on impact of hybrid performance. There are isolation standards that are varying from crop to crop. For best genetic purity, it should be compiled strictly and followed without fail. There are three types of isolation. First one is distance isolation, which needed to be maintained recommended isolation distance as per crop standards. Second is time isolation, which to be followed to escape flowering simultaneously within seed and other plots. Means there should be sufficient gap between flowering of two crops of same species. It will avoid the contamination. Third is barrier isolation, where the topographical surface features and natural obstacles are used. Now we will study in details about distance or spatial isolation. The spatial separation or spatial isolation required between a seed field and other sources of genetic and mechanical contamination, especially between varieties of cross pollination. In case of spatial isolation, for the extent of outcrossing is higher, we have to increase the distance. Here we will go through different crop hybrids about their isolation distances. In case of corn, generally the isolation distance is followed 250 to 400 meters depending upon the grain color. In case of pearl millet, the isolation distance is followed 200 meters. In case of sorghum, it is of 100 meters. In case of hybrid rice seed production, the isolation distance is 100 meters. In case of sunflower, 1000 meters or 1 kilometer is necessary. In case of hybrid mustard, it is of 1000 meters or 1 kilometer. And in case of castor hybrid seed production, it is of 300 meters. In case of higher class of seed, we have to increase the isolation distance. In case of breeder seed, the isolation distance is higher compared to foundation seed, and accordingly in foundation seed the isolation distance is more than certified seed. Means if the classes of seed is higher, breeder seed is the highest purity seed, so it is having the highest isolation distance, then foundation seed by certified seed. In hybrid seed production, there should be more wider isolation distance compared to varieties. Now to understand the importance of isolation. Isolation plays an important role in preventing unwanted cross-pollination, and it is the primary practice which is required to keep varieties true to type and avoid any type of contamination. Isolation begins with an understanding of the distance required to limit or eliminate chances of cross-pollination between two varieties of the same species. Isolation by distance is the most reliable method, which involves providing enough distance between a variety and any sources of contaminating pollen to ensure that the variety remains true to type. Fourth safeguarding measure is rogging in seed fields. What is rogging? Rogging refers to the act of identifying and removing plants with undesirable characteristics from agricultural or seed production fields on visual inspection basis. Rogue plants are removed from the fields to preserve the quality of the crop being grown. Rogging is important practice in seed production to maintain genetic purity of seed. There are different rogging stages which vary from crop to crop. The rogging should be done timely and prior to any contamination. Rogging is done from vegetative stage to harvesting stage based on many parameters as morphological and reproductive characteristics. The critical stages which are most important are vegetative stage, pre-flowering stage, flower stage and maturity stage. So rogging should be done without fail and strictly to remove all off types or rogue plants which are undesirable and unwanted. Rogging is important practice to maintain the genetic purity in seed fields where the off types are removed timely before any contaminations. The existence of off type plant is another source of genetic contamination. Plants differing in their characteristics from that of the seed crop are called as off types and its removal is known as rogging. The main sources of off types are described as first one segregation of plants for certain characteristics or mutations, means there are changes in the characteristics due to their genetic change. Second is volunteer plants from previous crop. Third is accidentally planted seed of other variety of same crop. And fourth is diseased plant. So the off type is consisting of all plants which are differing from seed crop. Off type plants should be rogged out from the seed fields before they shed pollen and pollination occurs. To accomplish this, regular supervision of trained personnel is required and it should be done timely without fail to maintain the genetic purity. Now, significance of rogging. What is the importance of rogging? Rogging at all stages of the crop in the field is an essential requirement to maintain the varietal purity as it was at the time of release or notifications. So, rogging is important to maintain the true characteristics of the variety. Sometimes rogue plants are not distinguishable. It is difficult to identify before flowering. Therefore, rogging should be done as early as flowering or blooming starts. The doubtful plants about which we are not confirmed that these plants are true or different, so it should be removed. It should be rogged out. The rogged plant should be removed from the field immediately after rogging and destroyed, as they may survive for a few days and may release pollen which may contaminate. So, the rogging is a very, very important practice of maintaining the genetic purity in seed field. Fifth safeguarding measure for genetic purity is seed certification. Genetic purity in seed production maintained through a system of seed certification. The principal objective of seed certification is to maintain and make available crop seeds of good quality to farmers. There is need of qualified and well experienced inspection to maintain the genetic purity while seed certification. To achieve this, qualified and trained personnel from seed certification agency to carry out field inspections at appropriate stage of crop growth is important. They also make seed inspection by drawing samples from seed lots after processing. The seed certification agencies verifies for both field and seed standards, and seed standards are confirmed by such agencies to get approval as certified seed. So the crop seed should meet with all standards which is fixed by the seed certification agency, and it should be released only after verifications. Sixth safeguarding measures for maintaining genetic purity is grow out test. Genetic purity of any variety is confirmed by carrying out through grow out test, which is known as GOT test, where the growing of progenies during off season in the field is preferred, and another method, lab testing, is done for electrophoresis for the verification of the purity. These tests are essential part of seed certification agency, which is needed for hybrids and high valued seeds. The grow out tests are conducted to determine genetic purity of a seed lot wherever it is prerequisite for grant of the certificate, and also on the seed lots where a doubt has been arisen about the genetic purity. So such lots are recommended for genetic purity testing if it is recorded that there are chances of contaminations. Varieties that are grown for seed production should be periodically tested for genetic purity by conducting GOT test to make sure that they are being maintained in true form. GOT test is compulsory for hybrids produced by manual emasculations and pollination methods, and for testing the purity of parental lines used in hybrid seed production. Genetic purity is always expressed in percentage grow out test, and the seed sample is shown in the controlled condition with the authentic sample, and genetic purity is determined on the basis of observations made on the plant morphological and reproductive characteristics. So here in case of grow out test, the seed is shown and it is going through observations till reproductive stage. So this presentation was all about maintenance of genetic purity in seed production. Hope this will be useful to all of you. Thank you very much. I have given here my YouTube channel details, Journey with Vijay Kumar Shrivastav. Having request, please visit the channel, subscribe it, and provide your kind and valuable feedback. Thank you.